probably halfway asleep the first time. So today we have Hope Hart is going to lecture for us, and Hope is a scientist at Syngenta. She's a technical leader for product safety at Syngenta. She received her BS in biology at UNC Charlotte and her MS in microbiology at the school to beat Duke last night. She's had a 20-year career at Syngenta. Her first 10 years she spent in insect control research, and she discovered Syngenta's latest GM trait. It's called AgriSure Duracade. It controls western corn rootworm. For the last 10 years, she's worked with Product Safety Group that assesses the safety of GMO crops. And the title of her talk is The Great Trait Debate, Is It Safe? Thanks, Hope.
Thank you. Thank you all. I'm here today to talk about GMOs. Have you heard the term GMO? Can you tell me what a GMO is? What's it stand for?
Genetically modified organism.
Excellent. Genetically modified organism. You are ahead of the game. There are many people out there who don't know exactly what that stands for. Just to show a hand, so I know what my audience is here today. How many people think GMOs are maybe worth having out there, or at least worth talking about and hearing about? How many people say, I don't want them in my food? Perfectly fine. I have lots and lots and lots of people. I'm not sure about it. How many of you know somebody who says, I don't want that? You've seen it on social media. You've seen it. Yeah. So what I'm here today is to tell you a bit about how we test the safety of a genetically modified crop before it goes on the market, and hopefully answer any of those concerns or fears or questions you've had or you've seen out there, and help you know how better to talk to your friends and family when you go back out this afternoon on GMOs and what they mean and how they're tested. Are they safe or not safe? How can you say they're safe? Why do you feel comfortable saying they're safe? That's my goal today. I'm going to start with a bit of disconnects, which is a really important topic when you talk about GMOs. There's a huge disconnect between, in people's knowledge, just in the fact of what is a GMO. That is a huge disconnect. And then I'm going to go into three main messages that will illustrate the safety of GM crops, and you can pretty much use these messages to answer any question on GMO safety that you might encounter. Now, I can't pull this up here. My Syngenta Fireball won't let me get onto YouTube. We get the big hand at work. Can't go there. If you go home and Google Jimmy Kimmel GMO, did anybody see the Jimmy Kimmel bit where they went out and talked to people about GMOs? It is funny and sad at the same time. So they go out to a farmer's market and they say, what are you buying today? Is that GMO? No, it's not GMO. Well, would you buy GMOs? No. Well, why not? They're not safe. You shouldn't eat them. They're bad for you. They'll cause cancer. Well, what is a GMO? Maybe it's mutated. They didn't know what a GMO stood for, but they had this perception that it was bad. They knew it was bad, but they don't even know what it is. There's a big disconnect between people hearing something and kind of jumping onto that kind of bandwagon and not even sure what it is. A GMO is a genetically modified organism. In this case, we're talking about genetically modified plants. It's where you've taken a small amount of DNA and you've added it to a plant, and now the plant has a new characteristic it didn't have before. Insect tolerance, drought tolerance, things like that. Eating natural. I went to a luncheon one day for Toastmasters, actually, and the lady I was sitting with said, well, what do you do for a living? Okay, here we go. Now the phone's going to start getting the really lively conversations when people ask me what I do for a living sometimes. And I told her what I did, and she said, I don't want you messing with my food. I eat all natural. Okay, what is natural? On the left side of the screen are the natural, the ancient varieties of the crops that we know and love and eat on the right side of the screen. Top left, this is watermelon. Top right, corn. Looks kind of like a green bean there. That's corn. That's teosinte, ancient corn. Banana. Eggplant, carrot, cabbage, kale, broccoli, this family. So if you're going to eat natural, where do you draw the line? What is natural? I promise you, the DNA from these plants has changed and is different from the DNA in these plants. All naturally, okay? It happens. DNA changing is normal. It's completely natural, normal part of every living organism that has DNA. DNA is safe. The Washington Post published this article in 2015 where Oklahoma State University did a survey. And they asked people, what do you want to see labeled on your food? Number one, at 85%, was country of origin for meat. They want to know where their meat comes from. Number two, at 82%, was GMO. They want to know if it's a GMO. Number three, at 80%, they want to know if their food has DNA in it. Okay, this is where find out if you know what DNA is. There you go. It should be funny, sad at the same point. Every living thing has DNA in it. You eat DNA all the time. There's nothing new about that. Okay? But people don't quite understand that, and so they think if you're putting DNA into a plant, it must be something new and different. No, it's DNA. DNA is DNA is DNA. DNA is only made up of 4 base pairs, A, C, G, and T. All of us have those 4 base pairs. Corn has the same, bacteria have it, animals have it. What makes us all unique is the order that these bases are put into. DNA is DNA is DNA. Labeling. This is a really hot one that I get a lot of times. Why are you opposed to labeling? Well, the FDA came out and said there is no legal requirement for labeling a GMO because they're just like the non-GMO. The only time the FDA requires labeling is if you have substantially changed the composition of the plant or you've introduced some new hazard, an allergen or a toxin, or if you've changed the taste, the look, the smell of the plant. If you've done any of those things, you have to label. If you haven't done those things, then consider the plants to be equivalent to each other. So what is the value in a label? There's your kiwi label. Natural, all natural kiwi. This is what the label would look like if you had to label everything. Okay? Words I can't quite read. Okay? Lots of acids in there, lots of scary sounding things in there, all natural. This is another big disconnect, the hazard exposure risk. Don't get hung up on the jargon of it. The concept is very important. Basically, this says you have to be exposed to something that could cause a bad effect for there to be a risk. Was it 2 summers ago, we had 8, 9 shark attacks off the coast of North Carolina? Me coming into contact with a great white, that would be hazardous to my health. That would cause a bad outcome for me. I didn't go in the water that summer. There was 0 risk of me dying from a shark attack. Okay? It sounds basic, but when people hear things, when you start hearing things like, you're putting a toxin into plant and we're gonna eat it and we're all gonna die. Well, okay, that's pretty extreme, so let's think about that. We're putting something into a plant that will kill an insect. Is it possible that we could be exposed to it? Yeah, it's possible. Will it cause something bad in us? No. It has no adverse effects against us. There's no risk. You're putting insecticidal proteins in plants and all the butterflies are gonna die. Okay. Could butterflies be exposed to this? Yes. They eat corn pollen, they could be exposed to this. Is it going to harm the butterflies? Nope. There's no risk. You have to have both in order to actually have a risk. And a lot of times, some of the grand, kind of scary claims that I hear, it's only one or the other. You're exposed to this. Well, yeah, but there's nothing bad with it. Or this is horrible. Yeah, but we're not exposed to it. So you have to actually have the both. Something bad could happen, then you have to be exposed to it. That's where you get your risk. My 3 basic messages. Here you go. Equivalent, well-tested, endorsed. GM food is equivalent to conventional or organically grown food. All of these three processes, genetic modification, conventional agriculture, organic agriculture, they're all processes and ways to produce food. Luckily for us, they all produce safe and nutritious food for us. GM crops are extremely well tested and reviewed. I had somebody tell me one time that these things aren't tested at all. I was doing a panel discussion at Duke. They had a course over there, kind of like this one, and there was another lady on the panel with me, and she said, they're not tested at all. They just put them out on the market, don't know anything about them. Well, I'm going to show you some of the tests that we do and the number of tests that we do to show you how well tested GM crops are. They're endorsed by major global science organizations. Okay, I get it. I work for a corporation that is selling these seeds, that is selling genetically modified crops. How do you know you can trust corporate data? How do you know you can believe me because I'm the one selling whatever the crops are? I get that. I empathize with that. I have that same little level of mistrust of someone trying to sell me something. You could throw out all the corporate data and you will still find data that shows you GM crops are safe from academics and government labs. So you don't have to trust corporate data if you don't want to trust corporate data. There's still data out there for academics and government labs that show GM crops are safe. First point, it's equivalent. Equivalent. You're starting with something safe, and then you're asking the question, did the safety change? This is a very important mind shift in what we typically think about with pharma and with chemicals. For example, a drug, a pharmaceutical. You'll hear on the news, this drug may have side effects of kidney damage, heart damage, liver damage, and all these terrible side effects. Therefore, you can only be exposed to this amount. They tell you what the side effects are for this high level, and then they tell you, therefore, your dose is this. You can only be exposed to this. Chemicals may have this side effect, therefore, you can only apply this amount. You can only be exposed to this amount. That's because you're starting with something that's already toxic, and you're determining how much you can be exposed to that toxin. GM crops, we are starting with corn. You're starting with something that's already safe. You're adding some DNA to it. Then you're asking the question, is this plant now as safe as it was before we added the DNA to it? Okay. We've added DNA. Is that dangerous, adding DNA? What happens to DNA? DNA and proteins, when you eat either one of them, they're broken down into their individual building blocks. And then your body uses them to build your own proteins and your own DNA. One of the challenges I hear is, you're going to eat this DNA. It's going to incorporate into your DNA, and you're going to start making mutant proteins. Well, there's a little bit of that that's true. The part that's true is, you will incorporate some of that DNA, but as these individual building blocks, not as actual genes that will result in a protein. Okay? If I took my laptop, I slammed it down, and I broke it into 1,000 pieces. If I pick up one piece, I do not have a functioning computer. Even if I scrape all the pieces together, I do not have a functioning computer. The parts have to go together in a very ordered manner to build something that works. The same thing is true for DNA. The bases have to go together in a very specific way to actually build a gene that could then possibly make a protein. Breakfast this morning. What did you have for breakfast this morning? Anybody? Anything, any bit of breakfast?
Biscuit.
A biscuit. You ate some wheat. You ate a lot of wheat DNA and wheat proteins. You are not making wheat proteins right now in your body. When you ate that wheat DNA and those wheat proteins, your body broke them down into the individual building blocks, and you probably will incorporate some of that wheat DNA into your DNA as the individual building blocks. The proof for that is you eat DNA all the time, and you do not turn into the organisms that you're eating. You are not turning into a wheat plant. It seems like a basic concept, but it gets lost when you start hearing the kind of grand scale scare things. This slide is intended to show you all the different layers that we test at. So when I say we test genetically modified crops, I don't mean we just look at the end product and say, yep, still a corn plant, That's good, put it out there. No, we do look at the end corn plant. We also look at what's produced inside the kernels. We look at what's produced inside the plant cells, the transgenic protein and the transgenic DNA, so the bit of extra that's been added to the plant. So we look at all these different layers to determine safety of a genetically modified crop. We conduct 80 to 100 studies on every one of our GM crops before they go into the market. Why should you trust corporate data? We work under quality systems that academic and government labs do not have to work under. We have internal systems that we work under, quality management systems. We work under good laboratory practices from the EPA, and there are also global guidelines that we follow, like from OECD. Working under the GLP, what we call them, under the EPA guidelines, it's kind of like doing your personal income taxes with the IRS standing over your shoulder. That's what it's like, seriously. We're audited every 3 years or so. It's just it's a standard rotation. We were audited a couple of years ago, and the EPA auditor came in, and he said, I want to see this study that was conducted 5 years ago. Okay, we brought out all lab notebooks to show him all the data so he could see everything. He opens up to a page and says, okay, it says here, you incubated this protein at 37 degrees for 20 minutes. How do you know it was 37 degrees? So then we go to the equipment logbooks from 5 years ago, and we say, okay, I see here that Susan Smith did the experiment, and there's her initials on the same date where she did the experiment and she used the incubator, and I see that this incubator number 8 had thermometer number 10, and thermometer number 10 had been calibrated the previous month to make sure it was working correctly, therefore we are confident it was 37 degrees. That's the level of detail we go through. It's not just a cursory look of, oh yeah, the study looks fine. No, they go into, you said this, prove it to me. Prove that is what you did. That's the level of kind of scrutiny that we work under, and the kind of auditing and oversight that we have. Our data is also reviewed by regulatory agencies around the world. They review our data, they ask us questions on it. Sometimes they may ask for more data. Sometimes they want clarification on something. Sometimes they produce their own data as well. So sometimes we have to give them materials. We have to give them seeds so they can grow their own plants and extract their own the DNA from those plants and do their own studies, or they want to do their own work. GM crops have been approved in over 60 countries. Okay, so our data gets reviewed around the world by 60 different countries and then approved in those countries. When you're asking the question, is a genetically modified crop safe to eat? There's 3 main areas that you're looking at. What is the potential of the protein to be an allergen or a toxin? And is the plant wholesome and nutritional? You started with something wholesome and nutritional. Did you change that in this process? Allergenicity. The source is the big one. Where did you take that DNA from? If a researcher has 2 traits that both give them drought tolerance, one came from watermelon, one came from peanuts, take the one from watermelon. We don't even want to deal with the peanut source. Even if it's not the peanut allergen you're working with, you don't want to deal with an allergenic source at all. Same thing is true for toxic sources. We do not take DNA from allergenic sources or toxic sources. Bioinformatics is used for both of these as well. This is where we take the protein sequence, just the string of amino acids, and we search that against all known allergens and toxins to see if there's any similarity in their protein sequences. This helps us try to figure out, is it likely that our protein would look like or act like an allergen or a toxin? Does it have structure similar between allergens and toxins? For allergenicity, we look at different physiochemical properties, digestibility. Digestibility, the idea behind this is, the longer a protein is stable in your gut, the longer your body has to mount an immune response against it. Okay? This is not 100% true for all allergens. Some allergens, very rapidly degraded. Some non-allergens, very stable. However, there's a link. There is a direct correlation between the two, so we use this as one of our criteria. If you ask me, can you promise me that that protein that plant is producing will never, ever become an allergen? The answer is no. Can't promise that. People become allergic to different things all the time. We can't promise that. What we can say is that there's no greater likelihood for that protein to be an allergen than any of the other 30,000 proteins that are in corn. So someone may develop an allergy to that at the same rate they would develop an allergy to the kinases that are in their plants, or their DNA replicases that are in the plant, all the other enzymes and proteins that are being produced by that plant. Toxicity is a little bit more black and white. We also look at source. We look at mode of action and history of use. Did this protein come from something that we eat all the time, or we've been exposed to, or is it something that mammals have never seen before? The cry proteins are a great example in how this mode of action history, both of these. So the cry proteins are crystal proteins that are produced from bacillus bacteria. Bacillus bacteria in all of our yards. Very common in dirt, found in all dirt supplies. If you've ever gotten dirt into your body, you have been exposed to these bacteria and these proteins. Okay. Well, it's not a part of our normal diet, usually, hopefully, dirt isn't, but it's not a part of normally eating. You have been exposed to it. What happens when you eat these proteins? One of the concerns I've heard is, if this protein is strong enough to kill this insect, it's going to harm my baby. My baby's a small organism, it's going to harm the baby as well. You need to know how these things work to understand, is that a true fear or not? Is that a real thing you should be concerned about or not? So what happens when an insect eats a cry protein? The insect eats the protein, goes to its gut, and it folds in the right shape. Okay, it gets folded in the right three-dimensional shape. Then the insect has a protein already that cuts it and activates it. That active cry protein binds another protein on the insect gut membrane, pokes holes in the insect gut. Okay, what happens when we eat the cry protein? We eat the cry protein, it goes to our gut. It does not fold in the right shape because our gut environment is very different from a rootworm gut environment. We do not make the protein that clips it and activates it. We do not make the protein that sits on our gut membrane that the cry protein binds to. So when we eat a cry protein, it gets digested or pooped out like every other protein that you eat. Okay? So understanding a bit about how these things work. On the surface, it can sound scary. If it kills an insect, why not a baby? No, there's no reason to think the two are connected at all because the insect is very different from the baby. Very completely different. We also do an acute oral tox study in mice. This is your black and white toxicity study for mammals. What we'll do is we find out how much of this new protein that we put in, how much is in kernels, because that's what a human would eat, a mammal would eat. How much is in kernels? We multiply that by 1,000, and that's what we feed mice. And you look for any adverse effects. Okay, so at least 1,000-fold higher than what an organism would actually eat in the environment, what you would find in the environment. And that is the standard go, no-go safety against mammalian study. One of the questions I get a lot is, the reason you say they're not toxic is because you're not looking hard enough to find toxicity. You're not doing the correct studies where you would find toxicity. These mouse studies are very standardized. They are the same studies that are done in the pharmaceutical industry and in the chemical industry as well. The difference is, in those industries, you do see a toxic effect on mice. They do the same thing. They take their chemical or their drug and they multiply it and they feed this to mice, and they do see a toxic effect. So then they keep coming down on the dose until they don't see toxic effects anymore. With corn, there is no toxic effect. Sometimes I think it might be easier for our industry if you could say, there's your line, don't eat that much. The reality is actually better than that. Reality is there's no toxic effect. We're not seeing any toxic effects with the same studies at 1,000-fold higher amount of proteins than what you would actually consume in a plant. Wholesomeness and nutritional equivalence. These studies also go back to the equivalent argument that I was talking about, the equivalent point. We do studies called compositional analysis and then feeding studies. The feeding studies are using broiler chickens and rats. It's where you feed them GM grain and non-GM grain, and you see do they grow the same. Are they nutritionally equivalent? Compositional analysis looks at over 60 different analytes in this plant to see if there are any differences between the GM and non-GM. It's equivalence. You look at things like sugars, proteins, fats, nutrients, anti-nutrients, vitamins, things like that. You're looking to see, Are they equivalent? And also, are there any unintended effects? These studies are very important for the unintended effects. We've put DNA into a plant, makes a protein, kills western corn rootworm. Okay. What if all the vitamin A left the corn plant? It was no longer producing the corn plant. That would be an unintended effect. We don't see those types of things. If we did, they would not make it out onto the market. That's one of the things you're looking for. That's why you do these studies. We don't typically see those types of things. When you're talking about safe for the environment, the agronomy studies are very similar to the compositional analysis studies, but they're on the more macro scale, more phenotype scale, if you will. You're looking at the whole plant at that point and saying, are these two still equivalent to each other? Have you ever heard the saying, if it looks like a duck, it walks like a duck, it quacks like a duck, it's a duck. That's kind of what agronomy studies are. Does it grow like a corn plant? Does it mature like a corn plant? Does it pollinate like a corn plant? Does it yield like a corn plant? It is still a corn plant. This combats that whole tomato and syringe. It's not a tomato and a syringe. It's still a corn plant. It started as a corn plant. It's still a corn plant. It seems kind of obvious to us, but we had a group of they were mommy bloggers. That's what their title was. They blogged about food issues, and they were all on the fence about GMO. And so they came to our site and BASF did a tour of the whole industry here in RTP, and I got to talk to them at the beginning of the day and the end of the day. So late in the afternoon, they, oh, we didn't understand, and now we realize we're not as afraid of GMOs as we were. We understand more about what they were. So I was asking, what did it? I go out and talk to people all the time about the safety of GMOs. What did it? What changed your mind? What made you feel secure? The answer was not my brilliant presentation. The answer was that going into our crop lab and seeing the plants. That's what reassured them about the safety of GMOs, because they had in their mind, they didn't start out as plants. They started out as something in the lab, some science voodoo, created this plant. No, it's a corn plant, and it's still a corn plant. So that actually had the biggest impact on them than anything else, is just knowing that it's still a corn plant. We also determined the levels of protein that are expressed in different tissue types throughout the life cycle of the plant. That's so we can dose non-target organisms. This is similar to that mouse tox study, except it's on the environmental side. We find out how much of this transgenic protein is in the pollen of our plant. You multiply that, and then you feed that to your non-targets. Your honeybees, your butterflies, your earthworms, other insects that live in that environment that aren't targets for the protein. Okay? There is zero tolerance for adverse effects on bees. Just get that out of the way. Zero tolerance for it. If a GMO has an adverse effect on bees, it would never see the light of day. It would not be out there. Okay? There's zero tolerance for that. By the numbers. Mentioned earlier, we spend about $150 million from idea to market. Okay, that's how much money it takes to get a GMO from idea to market. Every year, we're over $1 billion in R&D. We talked about the number of studies, over 80 different studies. It takes 10 to 13 years for something to go from an idea in a scientist's head in the lab to a bag of seed that a farmer can plant in their field. 10 to 13 years. This is my favorite number on the slide here, 3 trillion. 3 trillion is the number of meals eaten by humans that contain GM crops. 3 trillion. 0 is the number of people that have had adverse effects. Okay? 0. Endorsed. My last point. They're equivalent, they're well tested, they're also endorsed. Okay? This is if you don't want to believe anything that a corporation says, okay, great, that's fine. Go look at all the other data. I like to point this one out because it came from the EU. Not because I think they're better scientists, but because they're not particularly pro-GMO. The EU funded a decade of research on genetically modified plants. Okay? Their conclusion, the scientific research conducted so far has not detected any significant hazards directly related to the use or consumption with GE crops, genetically engineered, genetically enhanced crops. However, the debate is still intense. The science and the safety agrees, the politics do not. Okay? This one is intended to show you where some of the government-funded studies have happened around the world. So it's not just U.S. The U.S. government has generated lots of safety data on genetically modified crops, but also all of these other areas. Size of the circles is just how many studies have been there. So for example, 25 in the European Union, 29 in Canada, 5 in Australia, South Africa, South America. All over the world, governments have generated their own safety data on genetically modified crops and found them to be safe. This slide is intended to show you again kind of where safety studies have happened, and also it's a brief list of some of the organizations around the world that have come out and said GM crops are safe. The World Health Organization, Switzerland, that's a pretty big one. The Academies of Sciences, French, Germany, European Union, our National Academy of Science, the American Medical Association. Lots of very well respected and objective scientific organizations have come out saying GM crops are safe. If you go to supportprecisionagriculture.org, you can find a letter from 123 Nobel laureates to Greenpeace saying, lay off the genetically modified crops. There's nothing bad there. They're all safe. And they also have a listing of over 270 scientific organizations like these, a lot of the Academy of Sciences, that have come out in support of GM crops. I have a handout for you before you leave today with a list of these websites on that you can go check out if you'd like to find more information. The GMO Answers is one of my favorite right now. Mainly because it has a place for you to ask any question you want to ask. You're not limited to just what's on there and what the authors wanted to put on there. You can ask any question you want to ask about genetically modified crops. A lot of these others, the Pundit, Biofortified, Genetic Literacy, March Against Myths, very nice kind of layman terms input so that you can easy to understand, easy to take to your family and friends. If you want to know what the real dangers are in your food, go to foodsafety.gov. It is an FDA site, and you can sign up for an email alert every time there's a food recalled in our country. It's a little disconcerting because they come out almost every day. You get email at least one a day usually, but they're usually small recalls. They're not the ones that hit the news. But if you want to see what the real concerns are, that's where you go. And the spoiler is, you will not see one recall based on genetic modification. Mythbusters. So you guys should be able to answer these now. This is a GMO, this isn't. That's my favorite picture. I'm not sure if that's my favorite picture or the one where the corn plant and the fish head's coming out where the cob is. Kind of trying to imply if you put fish DNA into a plant, you're going to turn it into a fish. That's one of my favorite ones too. Seedless watermelons aren't GMO. That is a native trait project. Okay? That was generated through breeding, conventional breeding. Papayas are. Almost the entire population of papayas are genetically modified. Cotton t-shirts are. Like say, there's GMO in every one of the dollar bills you have in your pocket because of the cotton that's in there. Tomatoes are not GMO. Wheat is not GMO. That's a big question I get. Someone has gluten insensitivity. It must be because of all that genetically modified wheat they're eating. There is no genetically modified wheat. A few more numbers. The number of search results for GMO and cancer in 0.23 seconds. 16 million. That's how many results you get for that. The number of cancers caused by GMOs in over 20 years, 0. The number of search results for GMO and allergies, over 1 million. How many allergies have been caused? 0. The number of search results for GMO poison, 2 million. The number of cases of GMO poisoning, 0. So there's a very big perception out there, fear out there, without anything to back it up. So what I hope I'm doing today is at least getting you to think about it a little bit more and go look it up for yourself. I encourage everybody to look it up for yourself. You don't have to believe me, but don't believe that anonymous person on Facebook that posted something either. Go look it up for yourself. So in conclusion, my 3 main messages around the safety of GM crops. GM food is equivalent to conventional and organically grown food. They are very well tested, and they are endorsed by major global science organizations. Thank you for your attention. And what kind of questions do you have?
So I know a lot of you are good in class, but if you have some questions for Hope, hopefully she can hang around for a minute and chat with her.
I'll leave these with Dr. Edmiston.
If you don't get one, it's the website list.